So I will try to do it like kind of an interactive presentation. Let's see if it works. So at least half of you won't fall asleep. So let's see. So the topic of the lecture is how experience shape brain specializations, but more specifically, I would like to put forward two main ideas, so you can actually doze to sleep after this first slide. One is that this traditional way of thinking about the brain as a sensory machine, with having a conference for vision and have 7,000 scientists dealing with the occipital cortex, and then have a temporal cortex and auditory, and all this division of labor that is based on sensory, is wrong. Okay, I will try to argue an alternative, and this alternative is that different lobes and different brain areas have different computational behavioral tasks, and we can access them using any sensory modality. A little bit hint to this we could see in several lectures this morning, which was a mind-blowing session. I really enjoyed it and learned a lot. I hope you do. So it's again thanks to Ruth for putting together this wonderful team. But I will try to go one step further and not only suggest that this organization of the brain makes more sense, and the new data since Hubel and Wiesel that were accumulated by my lab and several other labs in the world support this notion that we need to rethink about how we look at brain organization and reorganization. But I will try even to challenge the notion of critical period. I will try to show you that usually when people used to think about critical periods, they think about unisensory critical periods. Okay, so they think about critical periods in the frame of reference as each senses and how it's developed and how critical it is on this sense experience. And I will try to basically show you that the entire organization, large-scale organization of the human visual cortex can develop very similarly with zero visual experience, okay? And that, I think, is very important to this field, as Professor Melsov was showing us, like in the previous lecture. Actually, you know, all the important things in life, and social development and social interaction is one of these important things. The other I won't mention here, but you can think for yourself, are all actually multisensory experiences in which the information flips between the different senses, and there is no point in thinking about development, about critical periods, about specialization without thinking about this as a multisensory experience, okay? So now you can fall asleep. No, not yet. I would just like to thank several, like the granting agency, especially we're very thankful for the European Union, but also with the RC program, but also to the McDonald Foundation from the US and the Human Frontiers, and mentioned people that I'm going to present their work here, mainly Shir Hofstetter, Zohar Tal, Galit Books, Tomer Bechor, Roni Arbel, Shachar Mandenbaum, Lior Reich, Elastra Amit, which is now in Georgetown, and some collaborators, my Parisian gang, Stan Dehan, Laurent Cohen, and José Sahel, and my Amsterdam gang, Guvitske, Zorobin, and Serge Dumlin. Okay, so let's start. So my lab main aim is to understand the principle driving specializations in the human brain and their dependence on specific experiences during development, critical periods, versus learning in adulthood. In the first part of the lecture, I will focus on nature versus nurture factors in shaping up category selectivity in the human brain. And when I talk about category selectivity, I'm talking about selectivity to faces, to reading, to body, imitating body images, and so on and so forth. And in the second part, if I will have time, I will talk about nature versus nurture factor in the other areas that are actually much more unisensory, like retinotopic areas, like primary visual cortex, like the homunculus, the S1, and so on and so forth, which are topographic. So that we have a topography which map neighboring areas in the world into neighboring neural structures, like V1, like the Penfield homunculus, and so on and so forth. And this was even more thought about to critically depend on unisensory experience during development, and I will try to show you some data on this. Okay, so the question is, what underlies the emergence of anatomically consistent specializations in the human brain? And functional brain specialization were considered to arise during evolution through natural selection mechanisms. So this is the time scale, and to constrain to specific unisensory modalities. I'm now trying to put forward the classic Hubel and Wiesel type, and one support to this is there is anatomical consistency across the entire people in this room and in different, continents and different places, that different specialization, like for ocular dominance, for category selectivity for faces, for houses, for reading, and so on and so forth, for body images, is consistent across all of the brains that are here, okay? And we think that this ocular dominance and face and all this activity arise through this, the development of this specialization to be critically dependent on unisensory, in our case, if we look at the visual cortex, but I will also mention auditory cortex and somatosensory during critical periods. And we will try to suggest that maybe it can be better describe this specialization and all this category selectivity, for example, as task-specific computational operators which are sensory modality independent. And the working hypothesis is that if the right technology and training are provided, we can ask whether both category selectivity and topography, like in retinotopy, homunculi, and so on and so forth, can actually develop in the adult brain and can develop with a new sensory experience that learned in the adulthood. Okay, so it can be a blind learning to interpret the visual space using sounds, like I will try to show you, or it can be an adult that learned a new sensory modality, like far sensing heat that he doesn't have during evolution, and see how the brain develops specializations to this type of new sensory experience that you never had during your childhood or during evolution. Okay, so why do we even have this weird idea? And this was actually the first, my first PhD work, and basically together with Rafi Malach and Udi Zari, we basically showed, we asked a very simple question. We know from Rafi's previous work that there is an area called LOC that is specializing is the human homologue of area 18 in the monkeys, and basically this is the area that is doing object recognition, okay? So we ask, first of all, in this work, is there a homologue in the somatosensory modality that also builds the geometry of the 3D shape, but this time from touch, okay? And then the second level of the question was, if we want to do cross-modal matching, or we want to correct something based on touch and vision and so on and so forth, where would we combine these two information? And based on the traditional models like Masulam and others, we hypothesize this will be somewhere in the middle, yeah? Maybe in the middle of the parietal cortex. And the surprising result was that the peak activity that is selective for shape analysis versus textures, versus noise, versus movement, versus imagination, and so on, so forth, was exactly in the same part of LOC, LO, and we termed it LOTV, which basically respond to both of them. But what is even more interesting is that when you listen to typical sounds that objects made, there was zero activity. So how can we try to sort this out? And we came up with this idea that maybe the brain is organized to, first of all, this area is actually an operator that reconstructed geometry of space, and you reconstruct it using vision very efficiently and very far. But you can also do it a bit less efficiently and only to things that you can grab and reach using touch. And so this brain area is actually a geometrical analysis operator of shape. It has nothing to do to vision. But to prove this, we will need to take people that never had visual experience. So this is how I started to get into the world of blind people. And some of these blind even didn't have their eyes developed, so they never experienced one photon of light. And when we repeat this experiment, we get exactly the same result. So basically, the peak activity for rendering of the geometrical shapes in the visual cortex exactly in the same area that is LO, even in individual level. And when they listen to sound is very associative thing, yeah? If tomorrow God, sorry for bringing God or whatever you want to call it, try to switch meows, and another thing I learned is that sounds of animals are different in different countries. So we said for a rooster, kukuriku. I don't know, how do you say in your own language? Yeah, kakaduludu, and you have all these weird things. But anyway, so if we are going to switch the meow and the kakadoodle or kukuriku, then in the beginning you would see this rooster doing meow, and you're like, what the fuck, yeah? And then after some time you get used and you hear the sound, you know you hear a meow, you know it's a rooster. But if you touch it, you know what it is. So this was the basis of this idea, and the question is, how much can we generalize it? So there is a few questions here. One, touch we learned, as we saw in the previous beautiful lecture, touch we use from day one almost, yeah? So what happens if I will teach you somehow to interpret shapes using a new sensory modality, but I will teach it to you when you are 70 or 60? Will we develop, and let's say we are only going to have 5 hours of training, will we still develop this specialization in the same shape operator, or that we used to call still today, you know, vision scientists are very difficult people, you know, and it's very difficult, so it's still resisting. They would still call it a visual area no matter what evidence you would put forward. Some change their view. Sorry, I didn't mean to sound so bitter, but anyway, all good. And so what if we, for example, take congenitally blind and teach them when they are 30, 40, 50, 60 to become bats and dolphins, to interpret sounds, and now the sounds are not associative, but they actually render arbitrary shapes and so on and so forth. Okay, so how can we do it? We can use the beautiful model of Paul Bacharita, which was the father of this field of sensory substitution, and he mainly took images from camera and put them actually in a dental chair and projected the topography of the visual world in touch, which makes a lot of sense. Now, we thought this is great, but it's a bit difficult in the scanner, and also the resolution is very limited. For example, on the back, you have the J&D is 4 centimeters. So you're really limited in the number of, and if you want to do face, for example, you will need to do over 1,000. It's impossible, okay? So we thought the auditory modality could be really cool, and also I was a failed jazz musician, so I thought, oh, maybe it's a good time to take back the saxophone and try to encode this in music. But before we did it, we had one study which was very interesting and actually gives us another case which starts to challenge this assumption that these very consistent specializations in the brain are due to evolution and natural selection and this visual, purely visual experience, okay? So this was actually the case of reading and the visual word from area. So first of all, reading, as you all know, is one of the greatest inventions in human history, and it was invented only about 5 to 10,000 years ago. So it's very recent time in evolutionary terms. And the question that Stanislas Dehaene and Laurent Cohen put forward, first of all, they discovered that part of this ventral stream that is specialized for objects, for faces, Nancy Kanwisher did a lot of work in specializing the extrastriate body area. There is an area that is very consistent across any written language and any form, visual form, to reading, okay? Actually, if you look at the variance across this selectivity, this is probably the most consistent area in all the category selectivity in the visual cortex. Uri Hasson was doing some work also on this notion and showing there might be some visual biases like foveal versus periphery. But so this is like a really difficult problem, because how can we have something so consistent, actually even more consistent in faces and navigation and so on and so forth, when there is not enough time in evolution to develop something that would, you know, be so specialized? So they come up with a brilliant theory, which is called neural recycling. And the idea was the following. But again, the bottom line is that usually people used to think about things as unisensory. So the idea was that this area was a visual area that have some characteristics made it really perfect to analyze reading. So what is these things? First of all, line junctions. It's really like line junctions to the configuration, and it's also like Rafi Malach and Uri Hasson and Yifat Levi showed, it really likes the fovea, right? So this conjunction of unisensory attribution made it perfect to basically be specialized to reading when it was invented couple of thousands of years ago, and also when each of us were introduced to it when it was 3, if it was a nerd, or 6 or 7, or so on and so forth. So it's a beautiful theory, and it actually survived a lot of the very interesting Neuron paper, and it survived. And the first time we met, I was like, it's an amazing theory, but we have these studies about object recognition. So actually, what would be the prediction with Braille? So there was a couple of predictions. One is just like LO, we will have in the parietal cortex, we have the hand area there, in the Penfield homunculus, and next to it we will develop a tactile homologue of this visual word from area for touch. Okay? And the second question is that if these guys never had visual experience, for example, they suffer from macrophthalmia, they never had any photon of light seeing, they would not be able to have this specialization and recycling this. So we said, okay, this is a great model. And another thing is that Braille have 0 line junctions. Okay, it's all dots, arbitrary. So we run this experiment, and you can actually, and just to mention again, all the important things are multisensory. If you think about it, and this is a bit speculative on purpose, but if you think about it, reading is actually the first weak sensory substitution ever invented, okay? So it was even before the times of Paul Bacharita. If you think about it, what is reading system? Is you take something that is phonological, that builds words and so on and so forth, and you embed this information into visual symbols. So you substitute phonology in visual symbols. Even more recent, the invention of the numbers, right? It's even less. It's like so this is, again, you embed quantity in visual symbols, and you even have the music notation, even more recent, that embed the heights of a pitch using a visual symbol. So this is just to mention, and then we come to Braille, which is a really hard sensory substitution, I would call it, because you really take a visual symbol. Well, actually what you do is you take a phonology, auditory symbol, you embed it in a visual symbol, and then you convert it into a tactile symbol, and then the blind that never saw or did so in his life now read using a different sensory modality. And now we run these MRI experiments, and you can already guess the result. The peak activity across the entire brain at the group level was exactly in the same spot. So the blind are the red, the sighted are the triangles, and this is taking out if this is sighted by reading or visually or blind using Braille. And what is also really interesting here is that if you remove the brain tag, the group tag, you cannot distinguish, and also a computer software cannot distinguish from which modality the person was reading. So it's on the individual, again, the variance across the various group is attributed to the function of taking a symbol and assigning phonology to it. Okay? But again, so this is great, so it doesn't matter if you read through this, but the next question would be, what happens if we will teach somebody in the adult brain? And what happens if we don't have these years, months of learning to read, if we do it in 5-10 hours with a new sensory modality? So this is where sensory substitution becomes very handy again. And we developed these 2 sensory substitutions. So, and I'm going to show you data from 2 sensory substitutions. So the idea again is that you have a camera, you render the information using touch or sound, and you teach the blind for 5 hours, 10 hours, 50 hours, then you scan his brain before, after, and you look for the specialization for each thing. Okay, so that's the plan. But before that, I need to explain to you a bit better what's sensory substitution. So we would show you 2 types of sensory substitution. One is minimalistic, it's called the ICANN, and it just converts distance to obstacles by vibration or sound. And we can have a virtual version in the scanner, and we can have a real version. You can have blind going in mazes, and so on and so forth. The second one is a holistic. It takes the entire visual image, reduce it to 2,000 pixels, and keep shape, location, and color information using music. And this is the iMusic. So first of all, let me show you what is it all about. It's a 1-minute movie in English, or actually you have subtitles here, and the second one is in English.
לא הייתי מגדיר את זה ככה, אבל זה הליכה, כמו שאת אומרת, עם הפנים קדימה, שאתה יודע לקראת מה אתה הולך. זה לא שאתה הולך על עיוור, מה שנקרא.
מאיר, עיוור מלידה, מכיר את המסדרונות המפותלים והצפופים של האוניברסיטה, כמו את כף ידו.
האתגר או הסיפוק הכי גדול שלי זה להיות כמה שפחות תלוי בסביבה, אלא לעשות הכל רגיל, כמו בן אדם נורמלי, שנוסע לעבודה או הולך לבנק או לסופר או לקניות, אז אתה רוצה לעשות את הכל לבד, זה נותן לי תחושה שאני שווה, שאני רגיל, שאני כמו כולם.
המכשיר הקטן שמאיר מחזיק רוטט בכל פעם שהוא מתקרב לעצמים שמולו.
פה יש לי פנייה שמאלה אם אני רוצה, אני יכול להמשיך ישר, אין בעיה.
לכאורה מדובר במקל נחייה משוכלל.
נמשיך, יש כמה פניות, אבל נמשיך ישר, יש בחדרים.
אבל הבשורה שהוא נושא איתו יכולה להגשים למאיר את החלום.
המוח, ברגע שאתה מלמד אותו להשתמש במידע הזה, דרך משחק או דרך חוויה של להסתובב, בעצם מהר מאוד בונה את התמונה הזאת של איפה יש לי מכשולים, איפה יש לי אנשים אחרים, איפה יש לי קירות. Now the second sensory substitution, as I said, try to capture the entire visual scene and several objects with their location and figure ground segregation. And here you can see the amazing team that actually, doing all this work.
A song is worth 1,000 pictures. Meet Danny. She's one of 250 million people navigating the world in the dark. These Hebrew University researchers helped her world become a little more clear. It's a common misconception that our eyes give us sight. It's actually our brain's visual processing allowing us to see. Professor Amir Amedi and his team invented iMusic, a device which turns a camera's visual data into a musical language. The musical language tells Danny about the objects in the room, where they are in relation to one another, and their visual characteristics. Despite being a prototype, the innovation has already received over $4 million in grants towards brain research from the European Research Council and led to a radical new theory of brain organization and development. Renew Senses, the researchers' startup company, is now working to take this invention public for the benefit of visually impaired people around the world. That's why we continue to innovate, because knowledge moves us.
Now, before we dive into the real part of the lecture, which is what happened in the brain, I just want to mention one last piece of the technology, and this that today we have this deep learning and natural neural networks machine vision that can actually recognize for you the objects in some cases. So we actually developed this ability both in the cane level and in the camera level, so you can build the actual sensory substitution image with different objects and colors and so on and so forth, but you can also get some verbal description of what's going on. Of course, if there is several objects, you will need some topographic rendering, which is more vision-like. In the case of the cane, it's more simple. It just tells you what it is in front of you and then give you just the distance, which is very difficult to do with neural networks using the eye cane. Okay, so this is where we hope to bring it to the blind community. Now, what type of neuroscience can we do with it? So first of all, before that, I want to I think people, how this algorithm works. I have like 2 minutes to describe to you how. So usually we'll take like 40, 50 hours. But let's see what can we do. I thought instead of showing you all the regular demos, we will do a social demo, okay? So first of all, I would not tell you anything about the algorithm. I will just play to you this diagonal line. So this is how black and white would sound, okay? Now I won't tell you anything. I will just draw a smile, okay? I want always to be a musician or a conductor. I was failing it, but I will play with it here. And let's draw together a smile. I will give you the sound. Sing with me. And let's do together a smile. Okay. 1, 2, 3, 4. Now let's do a sad face. Okay. You got it. This is how the algorithm works. So basically, what you see is in the y-axis you have a pentatonic scale. One pentatonic. Look at Bobby McFerrin. Lecture in TED, 2 minutes TED Talk show how universally everybody can understand the pentatonic scale and not any other scale. It's an amazing demo. He shows, anyway, he's also a great singer. And the x-axis is time. It sweeps over time. And when you combine these two axes, you can form the smile or sad face and so on and so forth, or even a face after 40, 50 hours. Okay? How do you do colors? Different pixels have different colors. Each color is donated by different timbre, and then you combine both of them and you get basically the colors by association. So this is more like the auditory system usually works with association. Okay, and then after, the first thing they blind learn to do is all the lines, what we did here for 10 hours, but then they start to do something that is called distal attribution. Wow. In the beginning, when they have these sounds, they think these sounds come from their ears, right? They play it to the ears. But after about 10 hours of working with this, they start to understand that the sounds, just a second, it's jumped to the next lecture, anyway, so they start to realize that the information comes from the objects, not from the ears. So this is called distal attribution. It's actually happening during development to all the senses, besides touch, yeah? But also touch for other people, but this is almost immediate. But here we can have like a way to really study it when it's emerging. Then in the next level, the next stage, they would go into, a more natural environment. So for example, here Sigalit, it's not the great quality, but she tries to find her shoes in her room. And now it's really a slow motion vision, and it's quite frustrating in the beginning. When you manage, it's really fun for the blind. And then we take this blind, and what I'm going to show you data from is from 3 different categories. Body imitation, just like we showed in the previous lecture, and also with a beautiful lecture by Kaisers, and reading that I mentioned, and also faces. So let's see. Now, before I'm showing you the selectivity to each category, after they learn and manage to differentiate between these 3 categories, let me show you what is shared by all this. So actually, what you see here is a still image that shows you the Heschel gyrus. I don't know. Yeah. So this is the Heschel gyrus. So this is A1. And this is the occipital pole. So this here is where V1 is located. And here is the ventral stream. What you see, as I told you, this is a slow motion vision, so it takes them about 3-4 seconds to build the object, let's say this bottle, yeah, and pick it like you saw in the movie. So what we can do is actually use a time to repetition in the MRI that finished to scan the brain in 1 second, and then look how this activity developed. And what you see is that what we did, we took a visual image from the camera and embedded it in sounds and music. What the brain is doing the inverse thing when it's learning. It's funneled information from the ear to A1, but when the subject actually builds the topography of the image, the activity goes into the ventral stream, V1, and also parietal cortex. And this is shared by all the categories. So in a way, this is already some kind of proof that actually what the visual cortex, and this was another example when you read a word, it's the same thing. So this was already one example which shows us that actually maybe what we called, and what you saw there is people with 0 visual experience again, okay? So the visual system is building this visual-like topography with 0 visual experience, okay? So first the auditory is doing fast Fourier analysis of the heights and of the volumes, but who is building the topography, just like from the eye, it's the visual occipitotemporal fixoparietal cortex. Now let's go back to the question, and the first thing we did was to study this question of reading and try to replicate it with a very simple design. And again, but the difference now is that we teach them the sensory substitution when they were 30 to 65 years old, and we only gave them less than 10 hours of training. And then the experiment was pretty simple. We gave them 3 types of stimuli. They were able to recognize the different colors. They were able to analyze these 3 shapes, and the 3 shapes was the Roman letters V, X, and I, okay? Why did we choose it? Because we wanted to push forward this concept of computational task. In one case, you do analysis of the color, you don't care about the shape. In the second, you do analysis on the shape and assign phonology to it, when you have a target of the V, for example. And in the third one, you combine a quantity to a symbol, just like you do with numbers. But all the physical stimulus are exactly the same, okay? So now we are basically run this analysis. And what is really cool is that while we were doing this experiment, there was a group in the West Coast from Parvizi that used echo and showed that if you do reading of letters, you replicate sound around a visual workframe area. But if you are giving them numbers, you get activity in the right homologue of this area, which is called the visual number form area. But this time we were being in touch with them and told them, first of all, we are able to replicate this in vision and also during the sensory substitution. So we actually suggest you call it a number form area and not a visual number form area. So basically, if you have the same physical stimulus and you analyze and assign the phoneme, you have the visual workframe area, so this is the replication. And then if you do analysis of the quantity, you get this. And actually now we have unpublished data, which did a bit more analysis because we were very careful about it. It looked really weird. But even you can have vector string areas around V3, V4 that are selected for the shape condition that we did not publish here. Another thing was the question of the mechanism. How can it be that in a few hours you develop this selectivity? So what we did was we ran a resting state analysis, just like Uri showed you, that what beautiful things you can do with resting state. So we actually, in a different study, showed that if you seed the entire like V1 and V2, you can, in resting state, you can basically map the entire retinotopic areas. Well, they don't see anything, and you can do the same thing with the congenitally blind individuals. So that's another indication that first of all, the system is reliable because you can map the areas even without visual stimulus, and it's the same. If you seed the hand area and the foot area, you get the other homunculi and so on and so forth. So what we did here was to map the resting state in these subjects both in the sighted and congenitally blind, and what we found is that if you seed, when the subject did nothing, the visual word form area, and you subtracted the activity in the number form area, you get selective connection into the phonology and the language system. And when you did the same thing in the number form area, the connectivity goes into the number quantity regions in the parietal cortex. So actually what we are suggesting is that the connectivity is innate to some extent, and each patch of the cortex have a different microcircuitry that is optimal to do shape operator or 2D analysis or line junctions, but then each area also have a unique connectivity that then very quickly we can take a new sensory information or new experience and reroute it into the neural network structure, okay? Great. So and then there is a more recent collaboration in which we actually took the almost impossible mission together with a Polish collaborator, Marcin Swed's lab, and teach sighted to read Braille, and this was much more difficult than teaching sensory substitution, but eventually, actually a blind instructor, they read using vision. They don't read using touch. It's really difficult. It's just on a JND, but they did a heroic effort, managed to teach sighted to read Braille, replicated the results, but then used TMS to show you can causally interfere with the activity and show that it's really a causal relationship. So it's really like a shape or task operators, okay? Okay, the next evidence comes from the extrastriate body area. You already saw that the mirror system is a very important system to our community. And I'm just joining to this community, so I'm trying to say, yeah. But anyway, the main thing is that you have to realize is that the main input into the mirror system, if you analyze, first of all, most of these examples that you saw are vision, right? Are visual examples. You see somebody copying and then you imitate. Now, the main input channel, and this is again NC Chemistry's work, is this area here, the visual extrastriate body area. And it's very important. For example, in the spirit of the World Cup that we just finished recently, we have these two guys, and one of them scored a goal and one of them missed a goal. Can you guess who thinks that this guy scored a goal or missed a goal? Can you raise your hand? Who thinks he scored? Okay, who thinks he missed? Okay, so it's about half-half. Now the second one, who thinks he scored a goal? Who thinks he missed a goal? Okay, I was actually tricking you. Both of them scored a goal. You can see here the image. But once you add the body information, using this visual extrastriate body area, it's very easy to judge. So we usually think it's all in the face, but no, there is a lot of social, and the same is missing, okay? So now, the question is, if we take this congenitally blind that never develop visual experience and so on and so forth, we put them, now we can use by the way, everybody showed here so beautifully that it's so early, during development, this ability to develop. I would try to argue this is also, I did a very serious study on this, and I try to argue this is also very consistent in evolution. I will show you this. It was a YouTube study. Anyway. And
you look like a little monkey.
So preserving evolution that even but then I found one example that didn't but like people scientists, I ignored it. But I started to get really angry emails from people.
Hey Jill, you know them old sugar daddy. They be tricking, they tell them girls. I said you can have whatever you like. I said you can have whatever you like. Yeah, sex on me. You sure know I.
Okay, now to put the joke aside, what about congenitally blind? They never saw. Can we teach them to do it? The answer is.
עכשיו למשל אני מבינה שזה אמור להיות קטגוריה של גוף מלא, ואני שומעת קווים אופקים, אז אני מניחה שהוא עומד ככה.
Yeah, and now we put her in a group of congenitally blind in the scanner. First of all, I can show you versus rest. You can see that the sighted recruit versus rest the extrastriate body area bilaterally with the lateralization to the right. It's documented in the literature. In the congenitally blind, you can see that is even more lateralized, and you get this extra activity in A1, which makes sense. But what is really cool in this image is that if you do a probabilistic mapping, which voxels in the brain respond in 100% of the participants, regardless if they are blind or sighted, you get this white preference exactly in the extrastriate body. And the connectivity story repeats. I don't have time, but it's the same, and also the peak, and so on and so forth. And faces, this is Marilyn Monroe, by the way. You can even have the lips in a different color, so different musical instrument. And this is the resolution of the eye music. So if you are perfect, you can get it, but of course it takes some training to get it. And basically, I don't have really time to show everything. I have only tried to start it late, so I have 10 minutes straight. 10 minutes is all I need. I would spare 1 minute for this because this is really cool. It shows how we train.
זה אף.
אוקיי, מאוד.
שומעים את שני הנחירה, את שני
הנחיריים? איך אתה שומע את הנחיריים?
כמו קליקים, מה זה גם? וואי, נראה לי שומעים את העיניים ואת האף.
יפה. ואתה שומע פה אולי עוד משהו?
שנייה. נראה לי שגם הגבות מופיעות.
מעולה, כל הכבוד. בוא תנסה עכשיו להגיד לי.
So this is exactly what you did, but this time with a lot of other noises, which is the rest of the face.
כל הכבוד.
Now, always after each lesson, we will have a natural session on real environment, like you saw with the objects and grasping. Now we will have a face session. And you can see here Ella and Miri, which are, she's learning actually the different facial expressions. So some of them she knows from birth, of course, but some of them she needs to learn, for example, surprise. Yeah. And now she would go in front of Ella and look at her and recognize that there is a circle there and she's surprised. And we will do the next generalization to a different subject and so on and so forth. What is really cool is that when we look on the fusiform face area, we see that not only we get maximum activity for faces and minimal for scrambled faces, and zero activity for voices, which do not contain this holistic shape information, we have intermediate activity for inverted faces. Okay, so they have a little bit of experience with upright versus inverted, and you can even have this medium activity there. So to conclude this part, and then I will have 5 minutes for the topographic part, our data suggests that visual cortices do not need visual experience to develop specialization. critical or sensitive periods do not depend exclusively on unisensory critical period, even if the cortex is more plastic. Now, we don't argue that the cortex is not plastic more in day 1 than in day 10 than in age 10, okay? We totally agree, okay? It's this dependency on this unisensory critical periods is that we challenge. And we suggest that category selectivity, and I will show you data for category selectivity seems to emerge independently of sensory modality. Now, and we suggest the mechanism would be both that the local secretary have a different computational powers, and also each patch of the cortex have unique connectivity pattern that help him to compute this sensory independent operator of the computational behavioral task relevant. So this is the mechanisms. I promised you a bit of data from the cane. So we have blindfold sighted navigating mazes in vision, sighted navigating blindfolded using the auditory modality, late blind, congenitally blind, and we found, first of all, that the peak activity for navigation is not in the hippocampus. It's actually negatively recruited in the human brain. So I don't know, this is interesting. The peak activity is actually in dorsal V6 retinotopic area, and this is now replicated in some other groups. And there is this beautiful specialization for navigation, but what is really cool is that the peak activity for blindfolded sighted, for late blind, and congenitally blind all happen in V6 only after they have a training that enables them to navigate in the maze, inside the scanner. So this is generalized also to the dorsal stream. So for the last 5 minutes, so the take-home message, also the dorsal stream obeys to localization and navigation independent of visual experience. There is literature, beautiful work, animal work, so this is not just MRI and TMS, okay? Steve Lomberg lab show exactly the same thing, just the mirror image in the auditory cortex of animals with genetic modulation that never heard in their life. And also there is beautiful work from Helen Neville's lab for sign language, but also recent work from Marcie Swen's lab on rhythms in the auditory cortex without auditory experience. And finally, I want to show you one really cool case study in which we ask the next question is that, okay, we have these high order visual areas that we talked about, the body area and the reading, and what about this concept that early on, all the senses are located in different places, and maybe these areas are really unisensory and you cannot develop topography in the adult brain, okay? So first of all, we had first hint back in 2003 and 2004 showing that actually what we show is that it's not working in the retinotopic areas. What we show is that V1 and other retinotopic areas are being taken over by language and by memory, and actually if you put TMS in this V1 area, you get mistakes for verb generation and so on and so forth. Marina Bedny took it since 2015 and had a beautiful work on language and math and so on and so forth, corroborating this. So it seems that it's only obey high order area, but then we actually thought that we never did the right experiment. The right experiments would be to take a congenitally blind and now study how whether the topography, the 2D topography of the soundscape is creating a retinotopic V1-like map in the brain if he learned it when he's 40, 50, 60, okay? So can we develop a whole screen that today we can look into dreams and into imagination using decoding methods into this V1, V2, V3? Can we have a screen like this developed for the sensory substitution? So we teamed up with Serge Dumlin, which developed, so first of all, I would like to mention that using this method, Fourier analysis method that I don't have time, but it's all published from our team in the visual modality by Brian Wandel, in our team by Eli, Noah Scharia, and Uri Hertz in the lab. We showed that not only you can map the Penfield homunculus in like 8 minutes, you can find actually new homunculi, like this lost homunculi of Penfield in the precuneus and some in the insula and so on and so forth. So it's very powerful. So it's actually much more widespread topography. But the second question is, can novel topography be formed in the adult brain? For this, we use the population receptive field method, which is optimal. You basically scan the entire visual field. I will skip this is just another demo. But we have a subject, and what you do in vision, you scan the entire visual field. If a neuron or a column has a small population receptive field, you will get activity only in a given area, and it will be very sharply tuned. If you have a large population, like in V2, and then V3 and V4, it will respond in much more location and much wider tuning curve. So when you do this, you get this beautiful two-dimensional map in V1, which gives you from the center to the periphery and all the locations. So based on this, actually, you can do decoding, and other people showed it in visual modality. You can reconstruct the image when the person is dreaming or thinking about the visual cortex, or when he's seeing something. Now we can do the same thing. We can do a sensory substitution. We took a blind that was the best expert in the eye music. We did the same mapping, but this time it was much slower, and it was soundscape that went into different directions. And what is really, really cool is that, so you see here, if it's a small receptive field, it's a very sharply tuned. If it's a large, it's larger, and you can find the location, and you will construct the entire thing. So first of all, when you run this on the subject, and you try to see his mind-eye when he's imagining this, without visual experience, this soundscape, you can see, first of all, the auditory cortex is very sensitive to it. I'm finishing. That's actually the two last slide. And you get this auditory cortex and Heschel gyrus, but you see variants also in V1 and all the areas around it, and also in ventral and parietal areas. So these areas are really sensitive to different locations and different sizes of the stimulation you're tested with. But now we want to see different locations. So first of all, this was a reality check. In auditory cortex, you get this beautiful mirror symmetry map of all the frequency, high, all this pentatonic scale, not to pure tones, even to music scales. And it's in mirror symmetry just like this. But when you look at the other areas, you can see that there is beautiful phonotopic pentatonic maps that developed in this individual, that is now an expert, that is in various places, and it encompasses the entire spectrum of the sounds, just like V1. Now what about time? Time is even more abstract, high-level cognitive. Also these maps can develop. You can see here a full map in the parietal cortex and LO cortex for the time dimension, which give you the x-axis. And when you put these two together, you actually get areas that are very similar to V1, in the sense that instead of having the two axes, the fovea, periphery, and the locations, it's one axis is the height of the sounds, the other is the time, and together they build the entire image. So the conclusion is that also global topographies, V1-like, can develop in the adult brain. And I will take it to new senses and some others, so but I think we can wrap up here. Thank you very much.
One question, if anyone has one.
Thank you so much for the wonderful talk. Can they actually use it effortlessly?
So first of all, the cane, and now the cane that have this capacity to recognize the love, and it's immediate. The other one is, it's a bit more tricky. There is a blind that really wants to use it. The problem is that right now, the learning is very demanding. It has to be done in the lab. And also we can, part of the regulation, you need to make sure they don't hurt at home. So there is issues of bringing it. But one thing they love now is that we put together the machine vision and the sensory substitution. So from day one, they already get the benefit from the glasses or from the cane. They know what's in front of them. And then they use this to guide their brain to learn what it is. And then this combination they really like. So this is very recently, and we hope to bring it, and we think that this is the right way to go. Yeah, so we hope it will be helpful also, besides like doing something.